Welcome, welcome, welcome. As always, it is such a pleasure annually to be able to welcome you to the Jeffrey and Joyce Myers Lecture Series. This is, I believe, the 10th such event. We've moved venues a little, but settled in here, and it's just lovely to see all of you here. It gives me great pleasure to introduce this year's speaker, Earl L. Smith III from the University of Houston. He is all Houston all the time, so if you want to say, gee, what other experiences have you had at other institutions, Earl will say none, other than the many that he's traveled to and spoken at and met people at. He has a distinguished history recently of having been the dean at the University of Houston College of Optometry for the last 15 years, which is a remarkable achievement just in terms of numbers. He's also served on the National Advisory Eye Council and been funded continuously by the National Eye Institute for 35 years, also a remarkable achievement. So longevity is clearly one of your strong suits. Persistence, tenacity. He's also served as a past president of the Association of Schools and Colleges of Optometry and president of the American Optometric Foundation. So those are just the facts, and you can read even more of his achievements on your program. But I go kind of way back with Earl. When I was a graduate student nearly 30 years ago, we both served on the American Academy of Optometry's Research Committee. And we served there together probably for almost a decade. And if I told you, besides the two of us, John Flanagan, who's the dean of Berkeley, was also on that committee during those 10 years. Tim McMahon, who's on the board of directors for the American Academy of Optometry. Tom Freddo, who was the director of the program at the University of Waterloo. So it was Carla and the boys, but besides that, it was sort of, turns out to have been a future dean's group. And what I remember from the little I remember from those meetings, no, that's not true. What I remember from those meetings is that we engaged in something that I think is unique in a career and yet something that all of you should seek. And that is, I felt like there was a lot of peer mentoring. So we all mentored others in our own life, or in my case, because I was a student, I was mentored. But I think we spent long hours debating the future of optometry, the future of the American Academy of Optometry, the future of our individual institutions. And Earl was a little ahead of us since he's been dean much longer than the rest of us who have been in that capacity. And I remember his advice and deep, clear, creative thinking about the issues that were really important. I think his research reflects that. And besides dancing on the deck in Westport, Massachusetts under the moonlight with this big group of people, clothed with this big group of people, and lobster dinners where we solved all of optometry's problems and most of the problems of the world, I consider Earl Smith a dear, dear friend. So without further ado, Earl Smith.
We don't remember much of that. But, we always talk about the good old days, and they really were the good old days. Okay, I have a title like I can talk about anything. Observations relevant to the treatment strategies for myopia. Before I talk about anything, I do have conflicts of interest. I have patents on any myopia lens designs, but they're not relevant to the talk. And I'm a consultant. I don't get any money from it, but I do consult on a couple of things. And I do get money from a variety of different sources. The long-term goal of our lab has been to try to reduce the impact of myopia. And in particular, we've been trying to find ways to slow the progression of myopia. And historically, our lab has looked at the optical performance properties of the vision-dependent mechanisms that regulate eyeball growth with the idea of identifying what controls eyeball growth and then turning that into, or translating that into a clinical management strategy to control the way the eyes grow. And so in the first part of my talk, I'm going to talk about the effects of ambient lighting on eyeball growth and refractive development. That's sort of an offshoot of what we've traditionally done. And then the second part of the talk, I'm going to talk about adenosine receptor antagonists and their potential as therapy for slowing myopia progression. And I have to admit, this is a relatively new line of research, and most of what I know about adenosine receptors comes from papers that Andrew Hartwig has written. So if you have any questions, Andy is going to answer. So, first off, you also got to talk about why this stuff's important and why do you worry about myopia. Well, it's a very common condition. It's highly prevalent. The prevalence is increasing. The severity of myopia is increasing. We know we are in the middle of a worldwide epidemic of myopia. That epidemic has largely run its course, at least in the urban areas of East Asia. There in the urban centers, kids graduating from technical high schools, anywhere from 80 to 96% of those kids are myopic, and the average degree of myopia is substantial. The projection models are particularly frightening. Some of the projection models suggest by the year 2050, half the world's population will be myopic. It's a good thing for optometry students to know. And unfortunately, a billion people will probably have high degrees of myopia, over 5 to 6 diopters. So that's a big deal. There's a lot of myopes, and they're big myopes, and the numbers are increasing. Myopia is expensive. It's expensive from an individual basis on quality of life issues. It's expensive from a societal issue. I mean, evidence on gross domestic product would suggest that simple uncorrected refractive error costs the world about $200 billion a year. And we know that myopia can limit careers, and it can certainly limit your ability to learn, especially when it's uncorrected. And we often don't worry about that in the United States, but we should. If there's a survey on urban areas on the East Coast, and 2/3 of the kids that need a distance prescription don't have that. And I find it unbelievable that happens in this country, but I can't imagine trying to learn without being able to see the board or anything else. But the reason optometrists need to be concerned about myopia is that it is a leading cause of blindness, and it's becoming more and more of a public health issue because of the increase in prevalence and the increase in degree of myopia. As this cartoon shows, myopia is commonly caused by the eye becoming too long. As the eye elongates, it puts the eye at risk for a number of different blinding conditions. And as you can see in this table at the bottom, high myopes, for example, are 40 times more likely to develop myopic macular degeneration than non-myopes. But the point I want you to get also from this lower table is even low degrees of myopia, 1 to 3 diopters, those folks are still at risk for these blinding conditions versus non-myopes. And what that translates to for us is that anything that we can do to reduce the eventual degree of myopia in our patients has potential long-term health value for our patients. And it's something we really need to think about. Now, with respect to actually trying to alter the course of myopia, the good news is, at least from, I'm going to give you a very biased perspective here, animal research has laid a very strong scientific foundation for a number of strategies that are beginning to show significant promise. And there's 2 particular observations from animal research that I want to emphasize, because I think these are the most important ones and the ones that have launched just a boatload of other research. I think without doubt the most important contribution of animal research is it's demonstrated without doubt that refractive error, eyeball growth, is regulated by defocus. In essence, the eye's effective refractive state. And this is a figure from a paper by Frank Schafel, and it is a pioneering paper because that idea was best illustrated in what are called lens compensation experiments. And this figure illustrates one of those experiments. It's a simple-minded idea. You take a lens and you put it on an eye, you alter the effective refractive error, and you see what happens. And what he showed is optically imposed refractive errors produce predictable changes. So you put a plus lens on an eye, you impose a myopic error, that eye grows slower than normal, it develops an axial hyperopia that compensates for the imposed error. The exact opposite is true if you put a minus lens on. You impose a hyperopic error, the eye detects that, the eye grows faster than normal, and develops a degree of myopia that is correlated to power lens. This observation, this lens compensation phenomenon, has been replicated in every animal species that's been studied in a systematic fashion. And we see this all the time in the monkeys that we work with. It also occurs in humans if you look at the right studies. Now, the second observation that I want to emphasize, I think it's actually one of the most interesting ones that come out of animal research, and that is we now know that the vision-dependent mechanisms that regulate eyeball growth are located in the retina itself, or in the eye itself, I should say. These mechanisms operate in a regionally selective fashion, and they operate independently of the rest of the nervous system. So the 500-pound gorilla is inside the eye. And once we realized that, that set up a race to identify all the components. Because each time you can identify a component, you can potentially intervene. And so what I've summarized in this box diagram on the right is some of what we've learned about the emmetropization cascade. We know that hyperopic and myopic defocus are the key signals that drive the loop. We know that in the neural retina encodes the sign of defocus. There are neurons in the retina that know the focus history of the eye, whether it's been myopically defocused or hyperopically defocused. There are a wide range of neuromodulators and neurotransmitters involved, acetylcholine and dopamine being probably those that are most prominent. We know that signal is transported across the pigment epithelium. It's not a passive process, it's an active process. We know there are changes in fluid and ion transport, and we know there are a number of cytokines and growth factors also in the RPE. The choroid is an interesting structure. The choroid changes thickness in a bidirectional fashion with optical defocus. Hyperopic defocus causes the choroid to thin and moves the retina back in the direction of the secondary focal point. On the other hand, myopic defocus causes the choroid to get thicker. In essence, the choroid changes in thickness will tell you which way the eye is going to grow. There's also a signal molecule, retinoic acid, that's bidirectionally modulated with respect to defocus. In the sclera, we change the biochemistry of the sclera based on focal history of the eye. For example, hyperopic defocus causes upregulation in the enzymes that degrade the extrascleral matrix. It also reduces the production of the building blocks, proteoglycans, for example, building blocks of the sclera. That results in a reduction in dry weight. The sclera thins, it becomes more extensible, and as a result, it elongates in response to normal interocular pressure. And so that elongation in response to that hyperopic defocus is the error signal that comes back, and this closed loop operates until the error disappears. And in this sense, defocus is the exact kind of signal that an engineer would want in a feedback loop. It's exactly what you'd want to run an emmetropization signal operating loop. The problem is we know that it's, from a visual signal perspective, it's more complex than that. And in that respect, the observation that has probably had the more impact on our understanding of visual signals actually came out of the Orinda study. And this is a figure that you probably have all seen. And what it shows is that outdoor activities greatly reduce the risk of children becoming myopic. This is, if you just look at it, this is a really strong effect, and it has not only practical implications, it has tremendous theoretical implications. And this one observation that's been replicated in laboratories across the world has initiated and spurred a lot of research, and the amount of research associated with trying to understand this observation has been increasing dramatically. And I'm going to show you a little bit about that in the first part of this talk. But this has been, when you think about it, a simple observation, but one that is really insightful and really does a lot for our understanding. But it shows us that it's not just defocus in a sense. So before I get into data, I will mention something about methods, about 2 seconds worth. I'm going to show you data from our laboratory. All that data is from monkeys. And we use the Cadillac monkey, the rhesus macaca mulata. All our primary endpoints, our 2 primary endpoints are refractive error nets, spherical equivalent, spectral plane refractive corrections determined by retinoscopy, axial dimensions. Most of them are A-scan ultrasounds, but we use some high-tech stuff as well. And we measure all this stuff longitudinally. We usually start at about 3 weeks of age for their rearing, and that's where we start our measurements. So we usually go to about 150 days of age. So let's look at why it is that outdoor activity reduces the risk of kids becoming myopic. And there are a number of ideas about this, and this is, I'll be honest, it's probably the most popular one. It simply relates to the fact that it is obviously brighter outside than it is inside. The light levels here on probably the desktops are probably maybe 150, a couple hundred lux on the front row. Not a whole lot more than that. But if you go outside today with a light meter, you would, on a day like today, you'd probably read 100,000 lux outside. So I mean, it's a bunch. It's 2 to 3 log and it's brighter outside. And so there are a lot of things that go on in the eye that are light dependent. I mean, I mentioned dopamine. We know that dopamine release, for example, is stimulated by light. The higher the light, the more dopamine release, and we know dopamine is involved in this cascade. So a lot of people started looking at the effects of light levels on refractive development. And Regan Ashby in Australia was the guy that really got this started. And I'm going to show you some of his results, but he showed, first off, that high light levels reduced the likelihood that a chicken would develop form deprivation myopia. I'm showing you data here from our monkeys that shows the same thing. All these animals were reared with a diffuser lens in front of one eye, and I should have mentioned this earlier. Anytime we put a lens on the eye, we're using a spectacle lens to avoid some confounding issues. But in the left side, our animals are reared under normal laboratory lighting, which is about 350 lux or so. And this shows the interlocutor differences. And what you can see, there's data from 18 animals over the years, and 17 out of the 18 develop myopia in the treated eye, and that's indicated by the eye bars of the data going down. Now, there's a lot of variability, and that just represents probably genetic differences in the eye's inherent growth rate, because form deprivation myopia is unregulated eyeball growth. We open the loop because there's no feedback signal, and so it's just whatever the natural inborn growth rate of the eye is, and so there's a lot of individual variability. But look what happens. We put the same diffuser on the monkeys at the same time, same age, same room, except we have some extra lights in it. We got 2 out of the 8 show myopia, but 6 out of the 8 show hyperopic shifts. And I'll tell you, these are large hyperopic shifts for monkeys. And what is key is we prevented form deprivation myopia in 3/4 of the animals, and we did this throughout the treatment period, and it was sustained as long as the animals were in the high light. Same kind of results we're seeing in chickens and tree shrews. They're actually more obvious in monkeys than they are in these other species. And it is light level dependent. The higher light level you go up, the more effect you get, at least with chickens. We haven't investigated that in monkeys, but it certainly has a huge effect in terms of form deprivation myopia. And the reason we think that's true is that light levels modulate dopamine. This is a figure from Ashby and Frank Schafel. And what it shows is the effects of the dopaminergic system on the protective effects of highlight. So this is the control, the white bar, down being myopic. This is what happens if you put the animal in high light levels. It reduces the degree of myopia. But look what happens if you put the animal in high light, you form deprive them, and then you inject the eye with spiperone, which is an antagonist of dopamine. You prevent the protective effects of high light levels. So this is a very strong demonstration that it's very likely that the protective effects of high light on form deprivation myopia come about as a result of light stimulation of dopamine. It's very obvious that dopamine is central to the phenomenon of form deprivation myopia. That may not be the case with defocus-induced myopia, and that may explain the results that you'll see in the next couple slides. This is data on the left from chickens, tree shrews on the right. And what it compares is the lens compensation phenomena under normal light versus high light. And for example, in the chicks, this is a 7 diopter lens under normal laboratory lighting versus about 3 times that, well, more than 3 times that much, over 15,000 lux. And you can see it does slow it down, but the eye still fully compensates. And in chickens, it slows down compensation by about 24 hours, about a day. In tree shrews, you see the same kind of thing. You get full compensation in 9 days. And unfortunately, this data from Todd Norton's lab stops here, but the data does go on. And by about another 9 days, even the eye that's in the higher light levels fully compensate. So at high light levels, you get full compensation. But it slows down in chickens and tree shrews. In monkeys, we don't even see an indication that it slows down. This is data from monkeys that were reared with -3 Doppler lenses in front of one eye, a normal light on the left, elevated light, 25,000 lux on the right. You can see there's a tremendous amount of individual variability in the rate. And in fact, when you compare growth rates of primates versus chickens and tree shrews, our growth rates are much, much slower. And so it's, I think, when you're given the small delays that you see with chicks and tree shrews, given the slow growth rates in primates, I don't think they'll be detectable in monkeys. And if they're not detectable in monkeys, they're probably not detectable in humans. That doesn't mean something doesn't happen in the time domain. I just don't think you're going to be able to see it. And that shows up, this is a vitreous chamber growth rate. I just want to hammer this point home. The black symbols come from high light animals, the white from normal light animals, and I'm showing this data just to show that it overlaps. And you can see on the right panel we're showing the average degree of relative elongation between the two eyes. There's no difference between the two groups of animals in this case. So the form deprivation experiments are pretty clear that there's something about high light that affects eyeball growth. And so the form deprivation experiments definitely support the idea that there's something about outdoor light levels that probably contributes to the protective effect. But the failure, at least this is our interpretation, the failure of elevated lighting levels to alter either the time course, at least in monkeys, and the final degree of compensation, and that's true in all species, that suggests that, in our minds, that defocus can override the effects of high light levels. But with respect to our cascade, it suggests we need to have a couple extra boxes here. Ambient light, the intensity has an effect, and I'm drawing a direct arrow up here to dopamine because high light levels modulate dopamine levels, and it has an effect when this loop is opened up. When you form deprive the eye can't detect defocus, and so you essentially break this bar here. And so there's no defocus signal in the loop, so the loop is, quote, open. And when you do that, ambient light can modulate dopamine, and that will modulate eyeball growth. This is a speculation on our part. We think pupil size, and optometrists should not neglect pupil size in terms of optics, but we have in this case. High light levels certainly affect pupil size. You get pupil meiosis when you go outside, and we all know that increases the depth of field, depth of focus. Now, pupil size has no effect on form deprivation because it doesn't affect the quality of the retinal image. I mean, it can improve it. But it can significantly improve the quality of the retinal image in an optical defocus situation. And so what we think happens in chicks and tree shrews is that pupil size reduces the strength of these defocus signals, and that's why it slows down. It eventually compensates, but it takes a little bit longer. The same delays may be occurring in monkeys, but we just can't see it because of the delayed time scale and the amount of individual variability in comparison to what you see in chicks and tree shrews. But ambient lighting is definitely an issue we need to take into account for a variety of reasons. Another popular reason to think about outdoor light and why it may protect kids from becoming myopic is that outdoor scenes have a different spectral composition of light than indoor scenes. And this is illustrated nicely in these figures from a paper by Folds et al. And what it shows, at least on a good day, and it's a good day here in Columbus, and this would have been true here, that short wavelength light, blue wavelengths would dominate the spectral composition of light outdoors. And this is a picture that's uncorrected for light levels, and it shows the spectral composition indoors from fluorescent lights. It's not color compensated. And what you can see is indoor lights, particularly fluorescent or tungsten lights, tend to be dominated by relatively long wavelength lights. And that's potentially important because the eye has longitudinal chromatic aberration. And what that means is that the effective refractive area of the eye varies with wavelength. the effective signal that drives eyeball growth varies with wavelength. And because of this, it's been hypothesized that outdoor light, outdoor time is protective because the eye is exposed to short wavelengths, and that one way to reduce progression of myopia is to reduce exposure to long wavelengths and to increase exposure to short wavelengths. And you've probably all seen, some people argue that we ought to use different kinds of lights indoors that we're using right now, and a variety of other things. We do know that spectral composition of light is a big deal in this emotropization game, because animal research has clearly shown that the eye can take chromatic signals associated with longitudinal chromatic aberration and decode the sign of defocus from that. So the eye can use chromatic cues from longitudinal chromatic aberration. It's probably not the only way to do it, but we know we can do that. And we also know that you can alter the spectral composition of light and alter refractive development. And I'm going to show you the results from a series of animal experiments, and this is where it sort of gets interestingly different. Anyway, this is one of the first studies that was done. This is from Frank Schaeffel's lab. And what he did is raise chickens in quasi-monochromatic light. And the mean data are on the right, and what it shows is that animals who were reared in blue light were more hyperopic than the animals reared in red light. And that's exactly what you would predict if you would assume that the eye grows to maximize contrast associated with luminance. Because longitudinal chromatic aberration changes the effective power of the eye, and the best focus for red light will be at a longer eyeball length than for blue light. And it turns out, this is a week-long experiment, and the difference in refractive error was a little over a 1 1/4 diopter, and they calculated the longitudinal chromatic interval between these two wavelengths at about 1.5 diopters. And so that match, they took as an argument that it's not likely that the eye uses chromatic cues, or at least it says that it can use luminance cues, and luminance cues can dominate refractive error. In fact, that sort of derailed us a bit. The direction of the results are right, but the story changes if you increase the duration of treatment. So here are data from guinea pigs on the left, chickens on the right. It's the same idea: relatively short wavelength light, longer wavelength light, relatively short, relatively long. The changes in refractive error are exactly the same as what Frank Schaeffel and his colleagues got. Is in the short wavelength light, the eyes became relatively more hyperopic. On the other hand, with long wavelength lights, the eyes became relatively myopic. But it was progressive in nature. It kept increasing and increasing. And so, for example, for the chickens, the predicted longitudinal chromatic aberration between the two wavelengths was a little over 1.18 diopters. But the observed difference at the end of the treatment period was over 7 diopters. And so this suggested that Frank's data, the conclusions were probably wrong simply because they stopped at the wrong time. It's just fortuitous. If they would have kept going, they would have probably seen the same thing. And so this argues against the idea that simple luminance contrast is going to run the game. But it also says that something's funny, because when you put an animal in quasi-monochromatic light, you take away chromatic cues. And so when you take away chromatic cues, things aren't working right. We got interested in this because color vision in our eyes, in primate eyes, evolved very differently from other mammals. Most other mammals are dichromats. We developed 2 photopigments in the middle and long wavelength area, and probably more importantly, we developed a midget system that compares signals in those 2 lines. And our color vision evolved very differently from what happens in birds. And so before we start talking about translating these kinds of results to the clinical setting, because these results would suggest that outdoor, spectral composition of outdoor light is a big deal. Our results, we thought we were going to get the same thing, and we got the exact opposite. But let me show you, set this up a little bit for you. This is we're halfway through these experiments. So I'm going to show you the data for red light. We're doing the blue light experiments now. This black line is the emission spectrum of the red LEDs. We basically installed an outdoor LED lighting system in one of our housing areas. We covered the entire ceiling. And so we can generate a boatload of red light. And then here's the absorption spectrum for the red, green cones, the blue cones, and the absorption spectrum for melanopsin. You can see there's plenty of light for red and green. There's virtually no light. There's probably an accidental absorption occasionally, but not much for blue. And there's a very little bit of overlap with melanopsin. But we know we certainly reduce the melanopsin signaling dramatically, because as soon as we put our monkeys in the red light, one of the first things we noted, and just by accident, in a sense, you look at the monkey, you go, those pupils are dilated. And so you put the monkeys in the red light, and they don't dilate completely, but they dilate by about a millimeter and a half relative to normal white light at the same light levels. So we did this red light room. We refurnished or redecorated, if you will. And then we reared 4 different groups of animals. Unrestricted vision or controls, animals without any helmets or lenses. Monocular form deprivation, monocular hyperopic defocus, and monocular myopic defocus. I'm going to show you the data from these bottom 3 groups first, and there's essentially 2 observations I want to show you for all 4 groups. The first is the effect of spectral composition on interocular differences in refractive error. So this is a busy slide, and the top row comes from essentially control animals. These are animals that wore diffusers to produce form deprivation or lenses to induce defocus, but they were in white light. So and this is the normal laboratory white light, not the high light levels. And the bottom are animals that were reared in the red room, the red light. The left, these are form deprived on the left. The middle is -3 diopters, and the right is +3 diopters. The point I want to hammer home is just look at the gestalt. In contrast to form deprivation and hyperopic defocus that consistently produces myopic shifts, we don't see that. Red light greatly reduced the likelihood that these animals would develop myopia in response to form deprivation or in response to hyperopic defocus. On the other hand, if we put a plus lens on an animal in the red light, they all consistently show changes in a direction to compensate for the imposed error. So we still see hyperopic compensation for imposed myopic defocus. The next thing I want to illustrate comes from the control eyes. These are, again, the arrangement at the top are the white light controls. The bottom are the 4 groups of red light animals, the unrestricted vision on the left, form deprivation here, -3 diopters here, +3 diopters here. But all the animals here, or all the eyes represented here, had a Plano lens in front of them. So it's unrestricted vision in a sense. And for comparison, these white squares represent the changes in refractive error that occurred during the treatment period. And these are for normal animals. And what you can see, there's a systematic reduction in the myopic direction. And that's the reduction in hyperopia that occurs with normal emmetropization. So the square bars represent the median changes in normal monkeys. And I should have pointed that out. These are changes in refractive error over the treatment period. So if you look at the top, there's variability, there's no doubt about it. But if you look, the data for the fellow control eyes of the treated animals in white light, they all basically show normal emmetropization. They all sort of cluster around the data for normal animals. But look at the control eyes in the red light. We have two out of 31, or 2 out of, can't remember how many it is, maybe 29 animals here is what that is. Only 2 out of the 29 animals showed myopic shifts that were equivalent to those in normal animals. 27 showed relative hyperopic shifts, relative to the median normal. And you can see most of them are substantially hyperopic, and they're progressive. They continue to progress in hyperopia as long as they're in the red light. And this shows data from the red light animals at the end of the treatment, and I'm very clever. The red light animals have red symbols. The black and whites are the white light animals. And I just want to emphasize here that at the end of the treatment period, Both eyes for the red light animals tend to be more hyperopic than the median normal animal. The dashed line is the median normal. And that's true for the lens-reared animals, the unrestricted animals, and the form deprivation animals are over here on the right because of the scale differences in order to get to see the data. But it's very clear that ambient red light prevents form deprivation myopia, it prevents lens-induced myopia, but it does not prevent induced hyperopia produced by myopic defocus. A couple changes that come about are associated with this. On the left, I'm showing changes in choroidal thickness. And what the gray squares represent data from normal animals. And the choroid increases in thickness by about 7 or 8 microns over the treatment period. But you can see both the fellow and the treated eye of the red light animals all became thicker. And 40 micron shift in a primate is huge. These are about as large a shift in choroidal thickness as we've ever seen. And as I mentioned earlier, the choroid seems to predict which way the eye is going. And so this suggests to me that we are perturbating this cascade in a way that has tricked the system. If you look at the characteristics of the eyes, these hyperopic errors look, for all intents and purposes, like the hyperopic errors produced by myopic defocus in white light. There are no alterations in corneal radius between the red light animals and white light animals. There's no changes in anterior chamber depth. There's no changes in lens thickness. The primary anatomical difference is the difference in vitreous chamber depth. And so if you look at in-treatment refractive error versus the ratio of vitreous chamber to the corneal radius, you get a really wonderful relationship. And so these are axial refractive errors. The constellation of changes look just like those produced by myopic defocus in white light. And so we think that, again, we're perturbating the system. So let me see if I've said all this. So narrowband long wavelength lighting results in progressive axial hyperopia, prevents form deprivation myopia, lens-induced myopia. And just by default, it says that blue cone signals are not necessary to prevent myopia. They're not necessary to produce hyperopia. And we'll also say that melanopsin signaling, that doesn't mean those ganglion cells are not involved, but melanopsin signaling by itself is not necessary to prevent myopia or to produce hyperopia. And it does say that chromatic signals are important. You think about this. So we're getting, the eyes are becoming more and more hyperopic. The amount of defocus is increasing more and more and more. And so defocus has lost the ability to regulate eyeball growth. At least hyperopic defocus has. And the fact that we get compensation for myopic defocus says luminance cues are still operational, at least for certain flavors of defocus. So why is it that red light does this? And I'm going to do some hand waving, but this is sort of our best thinking right now. I've drawn a myopic eye, and I've drawn it so that red light comes to focus on the retina, blue light is focused in the vitreous. We know that the eye can use cues associated with longitudinal chromatic aberration, and there's a variety of different kinds of cues, but let's just say the eye can look at the excitation levels of long wavelength versus short wavelength cones, so red versus blue, if you will. If it can do that and make that judgment, it can extract sign of defocus. So for example, in this condition in white light, the fact that red light is in focus, the photon density will be higher. If everything's balanced in terms of light levels, then the red should look brighter. At least the cone contrast should be higher for the red. Under red lighting, there really isn't a blue cone signal. The red cone signal is much higher than the blue cone signal. And so what we think we've done by putting the animals in red light, we think we fooled the system, that the eyes think they're myopic. And so they stop growing. So we think we've produced an erroneous error signal. And that's why it's progressive. And that's why the changes look like defocus. We think we fooled it. We think we fooled the system to thinking that it is a myopic system. And no matter what the eye does to change focus, that signal is still of the same sign. So, we put another box up here, spectral composition. And what we think it does, it influences the retina's ability to encode sign of defocus because chromatic signals are critical. We've taken the chromatic signals away and set up an imbalance that mimics certain kinds of optical errors. Okay. This is a huge effect. When we saw, we would love to be able to take a lens and produce these kinds of hyperopic shifts consistently. It's a really big effect. You would hope there'd be some clinical implications, but this is one where I think you just got to back off for a while. We just don't know enough about what the heck's going on. And let me just emphasize the contradictory results. Here we go. Long wavelength lighting results in axial myopia in fish, chickens, and guinea pigs. It produces axial hyperopia in tree shrews and monkeys. Long wavelength lighting does not prevent myopia in chicks. It does prevent myopia, lens-induced as well as form deprivation myopia in monkeys. I've looked at all the methods in these. There's essentially 26 studies on animals that have used various kinds of quasi-monochromatic lighting conditions. I've looked at all the methods. The intensities overlap, the wavelengths overlap. Onset and durations are hard to compare, but there's nothing obviously that stands out. I can't see a methodological issue that would explain this qualitative difference between chickens and guinea pigs and tree shrews and monkeys. My hand-waving excuse is that there are idiosyncratic interspecies differences. And I hate saying that because there's so many times that I've said that monkeys behave just like chickens, just like guinea pigs, just like tree shrews, that these mechanisms have been conserved across evolution. But color may be different. Our color vision system evolved differently, and that may be a true difference. But we're in the middle of the blue experiments now, and so it'll take us about another year to finish those, but that's something I'm really interested in trying to understand, is how the eye is doing this sign of defocus thing. The data are not better in humans. I've listed two recent studies down here, both from Japan. The Tori study claims that violet light suppresses myopia progression. That's based on some data obtained with intraocular lenses that screened out. One group had some that filtered out violet, the other didn't. But what you got to keep in mind is that these were phakic IOLs, and crystalline lens only transmits about 1% of violet light. So that signal's got to be really, really low. And the Zhao guys did sort of the opposite experiment. They gave kids blue blockers and compared them to lenses that transmitted blue and violet light, and they found no differences. And so right now the field's a mess, and you can find anything to support any clinical intervention you want to do. And so I think the only reasonable thing to do right now is not do anything. The reasonable one is get them outside. I mean, because regardless of what it is, it works. At least in terms of preventing things, there's a lot of debate about progression. I'm a believer that it actually affects progression too, but it'll be harder to see. But that's a different story. But anyway, we're a long way from. I think there's something there because the effects are so powerful. But you also got to keep in mind about how extreme some of the things we're doing. We put an animal in monochromatic light for 12 hours a day for. You're never going to do that to a kid, but you can change the spectral balance on an iPhone. So there may be some ways to deal with this. I can't leave the outdoor without, I'm a defocus guy, and I got to mention this. This is not a popular idea right now, but it's one that I really believe is important. This is a figure from Ian Flintcroft, and what it does is compares the dioptric topography of outdoor scenes versus indoor scenes. And the top is outdoors, the bottom is indoors, and in the right column, you can see that the outdoors is very uniform. And that's the key point. The dioptric topography of outdoor scenes is very uniform. You take an emotrope and you put them outside, the entire retina is in focus simultaneously at one time, and that is a very, very strong signal to stop eyeball growth. That's not the truth or the case indoors. And this is a picture by Neil Sharman, just to emphasize it. no matter where you look indoors, you're going to experience defocus, frequently hyperopic defocus, which we know is a strong signal for growth. The problem with identifying that is we know from animal studies that signals that stop eye growth are much, much stronger than those that cause it to grow. So they're much easier to detect. That may be why outdoors, the protective effect is so strong, so why it's been such a robust deal. But I still think we've got to figure out where it is about indoors that is not right. I think it's defocused, but we're going to have to still work on that one a bit. Okay, that's old stuff. Now new stuff. Adenosine receptor antagonists. I got to take a drink because I'm out of my wheelhouse on some of this. Caffeine and 7-methylxanthine, or 7-MX as I'll call it, are both non-selective adenosine receptor antagonists. 7-methylxanthine is a metabolic byproduct of caffeine. And there's been interest in these agents, or these antagonists, for treatments in myopia, and I'll show you the reason why. And there's been more interest in 7-MX because 7-MX does not cross the blood-brain barrier, in contrast to caffeine, so it doesn't have the arousal effects that caffeine has. So you can probably treat a kid without getting them to bounce off walls. The other reason that there's interest in adenosine receptor antagonists is that these are common things that are in your diet now. Common sources are coffee, and in fact I would argue that caffeine is an essential dietary ingredient. But cacao and chocolate, if you drink a Coca-Cola or anything with caffeine in it, you're going to end up with 7-MX and caffeine. And there's a tremendous amount of data that suggests that it's relatively safe unless you drink something like 30 cups of coffee. Now, it can affect your behavior, don't get me wrong, but I mean, in terms of side effects, these are fairly benign sorts of agents. Now, the initial idea about anti-myopia came from a study that Klaus Trier did on animals. And what he showed, and he had actually a good reason for doing this experiment. He recognized that adenosine was very much involved in the regulation of connective tissue. He recognized sclera as connective tissue. And so he fed animals orally 7-MX, and then he looked at their scleras. And what he found is there was an increase in collagen content, an increase in collagen fibril content, or diameter, rather size, thickness of the sclera. Changes that would presumably increase the strength of the sclera and reduce the likelihood that the eye would become myopic. So that was encouraging, plus the observation that adenosine receptors, they're located everywhere, retina, choroid, sclera, that adenosine receptors are modulated by visual experience. For example, you form deprived an eye, you can up and down regulate adenosine receptors. So that got people kicking about this, and let me show you a couple early observations that reinforce this. This shows data on the left from guinea pigs, data on the right from rabbits. These are normal form-deprived animals, and these are form-deprived guinea pigs that were fed 7-MX. And you can see there's a reduction in the amount of form deprivation, almost in half. In rabbits, there's a reduction. It's only about 18% or so, but it's in the right direction. These are data from chickens from Chris Willisett at Berkeley. Lens-induced defocus, minus lenses on the left, form deprivation on the right. Orally dosed 7-MX. I don't know how you feed a chicken 7-MX, and I'm glad I don't work with them. But what you see is 7-MX reduced the amount of myopia by about 22% or so. It did reduce the amount of form deprivation, but it wasn't significant. And these kinds of observations, and plus the anatomy that Trier had done, that encouraged him to do a clinical trial. This is the results from the only trial that's been published. And I just draw your attention to the 12-month data, because that's the only data that actually has a true control. This is the placebo group, and this is the 7-MX, and they were giving him basically 400 milligrams a day of 7-MX. And you can see that the 7-MX folks showed slower axial elongation, less amounts of myopia progression, but the results are not statistically significant. The subsequent data points are compromised. There's no real control there. But it was encouraging enough, and there were no reported adverse effects, that the Danish government allowed them to continue treating kids. And Trier has continued to add numbers to his database. Unfortunately, it's all observational. It's really anecdotal. It's not good data for determining efficacy. But he's got something like 400 or 500 kids now that are on current treatment, plus the ones he's done in the past. And he's learned, at least through his clinical experiences, that you need to do this twice a day instead of once a day. And we know that because 7-MX has really short half-life inside blood. So it's processed out of the body really quickly. And so he's doing some things that he thinks has increased in the effectiveness. He thinks he can do a lot better than this. But there are no clinical trials that show that this is real yet. But I am happy to report that there have been no adverse effects reported, so it seems to be safe. And so we decided to get into this because we thought it might be interesting. And so what we've done is we reared monkeys with either +3 or -3 Doppler lenses in front of one eye and plano in front of the other. So it's sort of one of our classic paradigms. We have two groups of experimental animals. In one group, we fed them oral 7-MX, and we're using 100 milligrams per kilogram, which is, I'll tell you, this is a very high dose. That's higher than, it's about 20 times higher than what the kids were getting. And we also want to do caffeine. And we want to do caffeine, but we want to do caffeine eye drops. We want to use caffeine because caffeine is arguably a stronger antagonist. The pharmacokinetics of caffeine are well worked out in the human eye. It's readily absorbed. It gets into the eye very, very quickly, and it tends to stay there. And so with caffeine eye drops, you can treat the tissue you want to treat without getting a lot of stuff systemically. And so we thought this might be an interesting way to do things. So we have 7-MX and caffeine. So I'm going to show you a bunch of data slides, and all you have to look at is the gestalt on these. These are controls. Normal light, -3 on one eye, and we've got data now for 21 of these animals. I'm showing you 10 representative examples. Here's the median of the group down here. Out of the 21, we had one animal that failed to show any compensating myopic anisometropia. But all 20 of the others did. And at the end of the treatment, on average, the degree of anisotropia was a little over 2 Doppler. So it really is a very consistent effect. And that's what I want to emphasize, is the red dots are always below the whites. And I should have hammered this home earlier. The black lines represent normal monkeys. So here's data from 7-MX, -3, 7-MX. Top two up here. These look like they're compensating, but they do compensate. There's a good myopic compensation here, a small degree here, but look at the rest of the animals. Essentially isometropic here, isometropic here, iso here, iso here. And then we have 3 animals where the treated eye actually becomes more hyperopic than the fellow eye. So the degree of hyperopic defocus is actually increasing. What a treatment for myopia. That's the deal. Caffeine basically looks the same. We have 2 animals that respond normally. They show myopic compensation. We have animals that show essentially no anisometropia. They do show hyperopic shifts in both eyes, so there's a hyperopic trajectory. And then we show 3 animals, or have 3 animals, that develop hyperopic anisometropias in response to minus lenses. So here's the opposite lens. This is the +3 Doppler lens. These are the controls, the Earl controls, and we've got 9 of these now. Every one of them shows hyperopic anisometropia that is outside the 95% confidence intervals for normal monkeys. And I just want to show that the red dots are always above the white dots. And on average, at the end, there's about 2 Doppler anisometropia that's in the right direction to correct the error. 7-MX and plus. Now, everything goes in the right direction. 5 out of the 6 show very clear evidence of hyperopic compensation. The only one that doesn't shows very large hyperopic shifts in both eyes and develops a very large degree of hyperopia in both eyes. Caffeine, same story. A lot of hyperopic compensation. One exception, there's a suggested compensation, but big hyperopic shifts in both eyes. And I just want to emphasize the changes in the next two slides. This is change in refractive error as a function of age. And again, the gestalt is what you need to see here. The gray bars are what you expect. There should be a myopic shift over time. But what you can see are both the treated eye and the fellow eye of the 7mx minus 3s, 7mx plus 3s. They're all on the top side of that. They're all showing these progressive hyperopic shifts. And as you can see over at the end of the treatment period, if everything shifted in the hyperopic direction, it's not surprising at the end of the treatment that both the fellow eyes and the treated eyes of these animals are more hyperopic than normal monkeys, and they're also more hyperopic than their lens-read controls. So this is caffeine, and the story is the same. More hyperopic shifts relative to normal monkeys in both the -3 and the +3 treated animals. And again, both the fellow eyes and the lens-treated eyes of the antagonist animals are more hyperopic than normals, and they're more hyperopic than, in general, they're more hyperopic than their lens-read controls. And just like we saw in red light, we see huge changes in choroidal thickness. This shows the treated and fellow eye of the 7mx minus 3s here and caffeine minus 3s here. This is 7mx and caffeine plus 3s, again, treated eyes, fellow eyes, hyperopic shifts, thickness shifts, increase in thickness relative to control animals. So it's, the choroid is showing thickness changes that are in agreement with the idea that the eye is going in a hyperopic direction. And if you look at the nature of the refractive errors, again, it's all vitreous chamber. Front of the eye is essentially unaffected by these manipulations. These hyperopic shifts come about because of a reduction in vitreous chamber elongation rates. And the exciting thing for us, in addition to the hyperopic shifts, is we've observed no adverse effects. Pupil sizes are normal. IOP, this caffeine is down a little bit relative to normals. I think when we get more animals, it's not significant, but I think as we get more animals, it will be. I mean, the reason people were originally interested in caffeine is they thought it might be able to lower IOP, and I think this is probably a real difference, but for normal animals, it's a percentage difference. So in terms of absolute millimeters, it's relatively small. So I don't know if that's going to hold or not. We'll see. We didn't see any surface abnormalities with any of the drops or anything else. We didn't notice any behavior. This is we deal with our animals a lot, and we bottle feed them 6 times a day. So we're mom, dad, and the world to them. So we interact with our animals a lot, and we have a socialization time where we bring about 10 animals in each group out and let them play together. And so we have a lot of opportunities to observe them, and we didn't see. And I'm not sure what a caffeined up monkey would look like, but to our trained eyes, we didn't see any behavioral difference. We did measure, serum caffeine levels. If you measure a normal animal, it's not detectable, and there's no caffeine in their diet. But if you in the caffeine eye drop animals, they're on average was about 200 nanograms per milliliter of blood, and that's about 20 times lower than the amount of caffeine that gets into your blood if an adult drinks a cup of coffee. So we're getting some caffeine absorbed systemically, but it's a relatively small amount. Now I have to admit that's a one-time point measure, and that issue needs to be worked out. Interestingly, we have not been able to detect 7mx in the blood. We're giving these guys massive doses of this stuff. And I'm confident that infants learn how to process this stuff fast. Their metabolism changes to get rid of it. I think that's why you're going to treat 7mx, you're going to do it multiple times. I think that's why Trier is trying to develop a time release thing for 7mx now that might work with kids. And the bottom just shows some OCT images. We didn't see any gross anatomical changes. We haven't seen any obvious differences. So we're encouraged that we're not poisoning our animals anymore. So we're excited. 7mx at caffeine, it increases choroidal thickness, just like myopic defocus, reduces overall growth rates. It reduces compensating myopia for lens-induced defocus as well as forehead deprivation. It promotes hyperopia in both in response to myopic defocus and in fellow eyes. That suggests to me that this has potential. I mean, there's a lot of issues that need to be worked out, but I think this is this has some potential. We don't know where it works. I mean, there are adenosine receptors in every major tissue of the eye. We think the action's in the retina, and the reason we think that is the shape of the eye looks like an eye that's been reared just with optics alone, the hyperopia. And it's all vitreous chamber, the front of the eye looks normal, and we're seeing choroidal thickness changes that suggest that we're perturbating the retinal mechanism, or at least we're perturbating maybe PE and choroid beyond. But our guess is we're perturbating retina right now. And I know Andy has some ideas about how adenosine can interact with both the acetylcholine system and the dopaminergic system that could potentially modulate the way the eye grows. So there are a variety of ways. But that's not a trivial thing just to brush off, because if we can figure out where it's working, we can really tailor the treatment to that site. But I'm excited by the fact that it's a robust effect, and it seems to be a relatively safe set of treatment agents. And I'd like to end by showing the folks that do things. I have Jenny's picture here because Jenny was involved with, we miss you. But Jenny was involved in all the highlight stuff. I'm showing Bhaskar Rumagan in the middle. Bhaskar was involved in a lot of this stuff. He's now at Cooper Vision. Bhaskar. I can say that. Shi and Krista Beach are relatively new to the lab. So is Lisa Ostrom. Li Fong Hung is, he and I are partners in this. We've worked together on this myopia stuff for over 25 years now, and he basically runs the lab. So anyway, thanks so much. I just got to tell you what an honor it is to be here. It's a thrill to be recognized like this. It's great to come up here and be treated really well. It's better. At home I don't get treated this way. And to see a lot of old friends and stuff. So it's been really great. Jeff, thanks so much for setting this up. I really appreciate it. But I will try to answer any questions.
Oh, all right. Any questions?
It just occurred to me, can all these things be done in vitro?
It's hard to get an eye to grow in vitro.
Oh, I didn't know that. So there's no feedback from the brain?
Well, you can cut the optic nerve. You can cut the best known parasympathetic and sympathetic inputs to the eyes, and all these mechanisms still work. The fact that you can manipulate parts of the eye, for example, if I take a diffuser lens or a powered lens and cover half the visual field, I produce refractive errors in half the eye, not the whole eye. It's hard to explain those kinds of things from cortical modulation or any other kind of modulation.
So the limiting factor is the eye growth.
The limiting factor is we don't know all the signals just right yet. I mean, we know defocus is important, but defocus is modulated by light levels and some other things. We don't know all the components, but we're learning. And there's a whole other realm of stuff when you start. Everything here is like we were talking about one point on the eye, but when you start adding the rest of the eye, there's other stuff that happens to it.
Right.
So no, this is not something that you can do in a dish. This is going to take animal models to dissect that cascade, and I think that's the value of animal models. But a lot of the hard work is going to have to be done by clinician scientists, because doing dose response functions in an animal can help define parameter space, but you can't extrapolate that to a human. You have to define that in human space.
And just curious, your form deprived
Yeah.
animals, they have amblyopia as well?
Oh, yeah, these are 22,000 amblyopes. I mean, we've done enough behavioral psychophysics on similarly reared animals to know that these are big-time amblyopes. Big time. We use, for clinical folks, we use Baumgartner occlusion foils, light perception occlusion foil, put it on at 3 weeks of age, and we'll leave it for 120 days. So it basically reduces your acuity to the very, very low end. Now, these are very severely amblyopic dogs. Now, the lens-reared animals, the ones that compensate are not. If they don't compensate, chances are they are. But amblyopia, there's a mistake in perception that amblyopia affects refractor. I don't think that's true.
Correct. But you can use the animal for both research.
Well, yeah, well, and we do. I mean, for example, a lot of the animals that I use in my research eventually end up in Yuzo lab, who studies cortical function in amblyopic animals. And in my wife's lab, who does behavioral.
You mentioned that you think that light levels are associated with myopia progression as well as myopia onset.
Yeah.
But you said we really can't detect it yet. So what makes you believe that it
Well,
is?
the
What makes
human
you believe
data,
we can't detect it?
the human data, you got some that say it does, some that say it doesn't, and the body, if you look at it all together, you're not convinced it. I don't think you can convince anybody it does. Why do I think it? I think getting a kid into the appropriate dioptric environment is important. And I think outside is important. And so I think part of progression is caused by a variety of optical conditions, and when you can avoid those, you go slow progression. So I do think it's there, but the data are obviously very weak at best. In contrast, in terms of incidence, they're huge, strong, really robust.
Yeah.
And it may be, once an eye becomes myopic, like you showed, 3-4 years before they become myopic, the shape of the eye starts changing. And peripheral hyperopic defocus may not cause myopia, but it may contribute to progression, particularly when you put a minus lens on there. Minus spectacle lens. We can argue about that.
At dinner, they can argue about that. Any other
Can I ask
questions?
one?
Yeah,
I'm so
totally.
sorry. I'm so interested. I know, right?
You don't have to apologize for asking a question.
I was
We're
curious.
just up here to
Well, all I can think of is that the development of the organs are regulated by homeobox genes. And I know that the development of the eye is regulated by homeobox genes.
Yeah.
And it would seem to me that why reinvent the wheel and just use the homeobox genes to do what they normally do? So do you know if light alters the activity of those genes?
Don't have a clue. And there are people that do know. I don't know. I know, Josh Woolman was sort of big into trying to understand, regulate organ system regulation like that, and he wrote this really, I thought, thought-provoking paper about this. There is something that seems to be different in a sense, and the problem that the eye faces is it has to adapt to its environment, and you can't predict what that's going to be genetically. I don't think you can predict the rules that will govern the eye to get to the right place, but you can't dictate what its dimensions have to be. But you can dictate the rules that govern the way it grows. And so that's how genetics sort of sets the gains of these mechanisms, and genetics establish this loop. But the loop needs to function to optimize shape. It's not just refractive; it's also shape. So I have to answer around the question. I don't know.
Thank
Sorry.
you. Thank you for entertaining that question.
All right, thank you very much, Earl.